First of all, thank you very much for the invitation, and particularly for the invitation to come here. And this is a splendid place. I've never been before, despite being born and brought up in Oldham, which is just up the road. I should say I particularly like the fact that they recognise the value of proper governance, because the mural over there celebrates the proclamation of weights and measures in 1556, which I think is great. Anyway, I'm going to try and answer three questions, really. One is, why as a pharmaceutical company are we interested in genetic and genomic data, and how does it help us develop medicines? I'm going to address a little bit why I think the UK Biobank resource is transformational and what led us to invest in it along with Regeneron to generate the exome sequence data. And then I've got one slide at the end which is about the future and how things might develop. So this has been touched on by Andrew and Jeff, actually. Why is genetics useful in drug discovery? And I'm going to pitch it in a slightly different way, just by giving you an example. Statins have been talked about. What statins do is reduce levels of HMG-CoA reductase. So that's something in the bloodstream. They reduce it. That leads to reductions in LDL cholesterol, bad cholesterol colloquially, and that leads to reduced cardiovascular risk. Now, there are genetic variants which essentially do the same thing. You know, they reduce HMG-CoA reductase, they reduce LDL, and they reduce cardiovascular risk. So in some sense, they then mimic what a randomized trial of statins looks like. And people often use this kind of picture to illustrate that if I was running a clinical trial, I'd take a sample, I'd randomize, so I'd toss a coin essentially. Half the people would get a statin, half the people would get a control. We'd run that forward, and in the statin arm we'd get reduced LDL and a reduced cardiovascular event rate. The randomization there is key because that's what ensures the two groups are comparable, that there's no hidden biases between or differences between those two groups that might bias the results. If we think about what genetics does, and this is sometimes called Mendelian randomization, we have a population and everybody in this room got half the genome from their mother and half from their father. Another way of phrasing that is to say that your mother gave her half her genome and your father gave you half his genome. But that was done at random. It's a random process. So you have this analog of randomization, which again means a group with the, let's say, normal genotype or genetic type, which has the normal levels of LDL and the normal cardiovascular event rate, should be comparable in all other ways to the group which got the beneficial, in this case, genotype group, which leads to lower LDL and a lower cardiovascular event rate. And that allows you to say, okay, and if you actually look at the results, they actually mimic each other quite well. And that allows you to make conclusions like the one Andrew drew about diabetes risk in statins, which is slightly elevated. So this is using genetics as a tool to almost anticipate what a trial might be if we had a drug. And that's really useful because trials are phenomenally expensive and genetics is quite expensive but much cheaper than trials. So how are we going to use this? The most obvious way is several people already mentioned, which is we could use it to understand existing drugs. The statin example is like that, for instance. Here's another one about GLP-1R agonists. These are widely used, becoming widely used for type 2 diabetes. With diabetes drugs, there's always a concern about cardiovascular risk, and a few years ago we found a rare variant that seems to mimic the drug mechanism of action in the same way as in that statin example. So then we looked at what the variant does, and this is the result of a lot of work by actually one of my colleagues, Robert Scott, who's in the audience. And I'll just explain this picture. The horizontal So we've got lots of diseases here. Horizontal line up the middle indicates, normality in a sense, the risk in untreated individuals. If you're this side of the line, reduced risk, this side, increased risk, and I'm comparing the genotype group, the group of individuals with the genotype that mimics the drug. So these are the people who have the drug mimicking genotypes here, and you see a reduction for type 2 diabetes. We knew that because we knew the drug worked. We see a reduction, reassuringly, in cardiovascular risk. The central points here are our best guess of the estimate. The line indicates the measure of uncertainty. So we're pretty sure the estimates are where the line is, and our best guess is in the middle. For nothing else can we really say anything at all because there's too much uncertainty. Incidentally, large studies have now more or less got the same, not large clinical trials have got the same answer as this. So that's great. The other thing we'd really like to do is do all this before we have a drug at all, so that we make decisions on the things, we move forward the things which are most likely to succeed. And we published a paper a couple of years ago showing that drugs with human genetic information are twice as likely to succeed as those without. Now that's a big deal if I tell you that the average cost of getting a drug from the start of the process through to patients is more than $1 billion US. So any improvement rate we can make here is really useful. It really matters. The other thing I think that people often don't know is that only 3% or 4% of drug discovery projects make it all the way to the end of the road and get to patients. So 96 or 97% of them fail at some point. So this is anything we can do to get that number down is incredibly valuable. So why is UK Biobank transformational? This is the picture I just showed you that I said my colleague Robert Scott put together. That took 1.5 years and more than 1,000 emails because we had to go out to researchers all around the world to essentially get their data on type 2 diabetes and CHD and all these other things. In some cases, we had to actually measure the genetic variant that we were interested in those individuals. We did the same analysis in UK Biobank recently. That took 1.5 hours and 3 emails. So this really is transformational. And it's not quite perfect. We have a bit more uncertainty here. You can see the lines are broader, but the qualitative pattern of the results is exactly the same. So this really enables us to do this right at the start of the process when we're really thinking about, is this a good drug target? Should we go after this one or that one? We can use this to make decisions. Just something about the future in my last minute. What's coming next? So we talked about the exome sequencing. The real value we see in that is, and Jonathan talked as well about, the amount of the genome that's captured already in UK Biobank, and it's quite a lot. The bits which are not well captured tend to be the rarer variants. So we were quite lucky. The type 2 diabetes variant that we were interested in was actually quite well measured. Many others will not be. And that's where the value of the exome sequencing comes in, that it really gives you all the base pairs in that particular slice of the genome. The other thing that we really need to work hard on is understanding how those variants affect disease risk. Because it's fine saying, here's a genetic variant, and it makes you a bit less likely to get diabetes, great. But can we fill in all that white space in the middle? How exactly does it do that? How does it act? And many of the things that UK Biobank has coming up are really going to help in that. The blood biomarkers, maybe those variants act by changing levels of a particular biomarker, that's useful. Maybe we'll see something on the imaging that helps us understand how that works. The other thing that UK Biobank is, of course, great for is that we're often interested in progression of disease. So the people, we don't treat completely healthy people for diabetes. That would make no sense. We treat people who have some early manifestations of disease. So we're particularly interested in genetic variants which tell us about progression. and that's telling me to shut up. So the only thing I'll finish with is I'm a statistician by trading, so I'm kind of contractually obliged to say that no study is ever big enough. And we really do need to, think about how we're going to replicate some of the findings in UK Biobank, maybe by analysis across multiple biobanks and cohorts. Thank you very much.